Well, thanks. It's good to be here, and I really appreciate the invitation. I've been looking forward to this conference for a while. I'm actually a poultry guy. I'm not a swine guy, although my first 5 or 6 publications were with pigs. I did my master's on pigs at Purdue, but I switched to poultry mostly because I'm lazy, and it's a lot easier to work with little chickens than it is big pigs. For those of you that knew me back then, I always had this antagonism between science, which I really liked, and really screwing off and having fun and racing motorcycles and that kind of stuff, which I really like too. And it's still my philosophy. You can do both, you just have to work a little bit harder. So for you graduate students, don't give up the fun things. Keep them going, but you gotta get serious every now and then. So sometimes I had some difficulty getting serious, but there's some good people like Tip Klein who helped straighten me out in those regards. So I appreciate that, Tip. As was mentioned, I grew up near here. I actually grew up just a little bit south of Greenwood, Indiana, so not more than about a 45-minute drive if there's no traffic, and really enjoyed this part of the country and still do. I get back quite often. But now I'm in California. I've noticed that when I come to the Midwest, California doesn't have an especially good reputation among people. It's referred to as the land of fruits and nuts, and I'll report back that it is, and it's getting fruitier and nuttier every year. I live in Davis, California, which for those of you who don't know, California, it's in Northern California. It's about an hour and a half if there's no traffic, northeast of San Francisco. Okay, so I'm going to talk today about inflammation and immune responses and some of their costs, nutritional costs especially, and a little bit about some mitigation rationale, although I won't be able to give you as many solutions as I would like to. So that my outline is I'm going to start off by talking about the performance and nutritional costs of systemic inflammation. When I say systemic, I mean away from the epithelia, so not in the intestines. I have another talk I can give in some future year on intestinal inflammation and some of those costs, but they're quite different in the way they're manifested. A little bit about cellular priorities for nutrients, especially when it comes to the immune system, relative to tissues like skeletal muscle, and again, a little bit about mitigation strategies. The general idea that's been out there really for many decades now is that the immune system needs nutrients to do its job. Cells have to proliferate. Cells have to make things like antibodies, various protective proteins, and there's got to be a nutritional cost for that, and that's got to be being drained away from things that we like producing meat or milk or eggs, reproduction, and the reason that theory's been around for a long time is that we've known for a long time, since long before anybody in this room was born, that when animals get sick, they don't grow. Basically, the growth slows down, and even if you force feed them to eat what they would normally consume when they have an immune response going on, they still don't grow nearly as fast. So it's not just a lack of appetite, although that is a big part of it. It's an inefficiency that occurs. And again, the presumption over the years has been that the immune system needs these nutrients. It's a high priority thing because it keeps the animal alive, and so it gets those nutrients and diverts them away from productive processes. So a little bit about inflammation, and I really don't want to spend much time on the mechanistic aspects, but I just want to give you a real quick helicopter overview. Inflammation occurs when the innate immune system, cells like macrophages and neutrophils, recognize patterns on things they encounter that look like microbial molecules. And these microbial associated molecular patterns are specifically recognized by groups of receptors called Toll receptors and Nod-like receptors, and there's several other families that alert the macrophage that there is something foreign there, that there's molecular signals of something foreign. And that by itself causes a little bit of inflammation. But there's another group of receptors that are on innate immune cells called danger recognition receptors. And these danger recognition receptors look for damage to host cells. They look for molecules that should be inside cells that now are all of a sudden being seen outside cells, or especially cytochromes, for example, that should be inside mitochromes or mitochondria are now floating around in the cytoplasm or floating around outside the cell. When they see that, that indicates damage. And that itself causes a little inflammation. If you combine these two things, you get a really large synergism and you get robust inflammation. And there's a third area that's just developed in the last couple of years, and that's called tactical infectious patterns. And this is a change in the amount of various metabolites that are inside the cells that it's abnormal. And homeostatic mechanisms can't return those metabolites back to normal. And so that's called a tactical infectious pattern that's detected by several different transcription factors, and that's an alert to the cell that there's something screwing up metabolism, maybe consuming nutrients, like maybe a bunch of bacteria, or maybe a virus that's taken over the cell machinery and changed the way nutrients are being used and produced. And so that's a whole other set of signals that then, if it is coupled with these other two, you get extremely robust inflammation. So again, any one of these causes a little inflammation, any two of them causes a lot, and all three of them causes a whole lot. Okay, and then when the inflammation occurs, cells like macrophages and T regulatory cells produce a bunch of cytokines like interleukin-1, tumor necrosis factor, IL-12, that then go out and regulate the inflammatory response both locally but also throughout the body. And there's other cytokines that can be anti-inflammatory, like these over here, and they would normally be produced except when you have these inflammatory signals. So that's the depth of immunology I'm going to go into today. There we go. I'm going to start getting back now to performance things. And some of this data is going to be a bit old. I have to apologize, but I bring it out and I rethink about it every now and then and come up with new conclusions. So if you heard this talk back in 1990, I'm going to present it completely differently and come up with different conclusions with the same data as I did back then, because we know a lot more about what's going on. But here's an example of a couple of experiments that my graduate students did where they injected either live or dead Staph aureus, and this is being injected into broiler chickens that during a fast part of their growth, they're about 10 days old. They're doubling their body weight about every 3 days, so it's a pretty robust growth model. This is live E. coli or dead E. coli, and this is the lipopolysaccharide from the cell wall of E. coli. And you see that really, in the case of E. coli, it really doesn't matter if it's dead or alive, or even if it's just a subunit of the cell wall. It all causes oops, I lost my, here we go. Oh, I'm screwing up. Sorry about that. It all causes a decrease in growth. So it's about a 30% decrease in growth. We're injecting this systemically. We're actually injecting it intraperitoneally, and fairly substantial decrease in growth. Staph aureus about 30% when it's live, not quite as much as when it's dead, but again, you get the feeling for the model system. We also sorted out whether or not this decrease in growth was due to food intake or if there are other inefficiencies. We did that. This is a control animal that's not challenged. This is one that's been challenged with E. coli, dead E. coli. There's about a 30% decrease in growth. This is a group that's not challenged but fed the same amount of food that was voluntarily consumed by this challenge group. And here the decrease in growth is only about 9.5% or so. So about 2/3 of the decrease in growth is due to food intake, and about 1/3 is due to other things that are contributing to the inefficiency. So graphically, it looks like this. So if I was smart, I would be studying the most important part, which is food intake, but I'm not. I've been working for 30 some odd years on this part, the 30%. So I want to spend a little bit of time on that 30%, because actually from a nutritional point of view and how you would think about feeding animals to help rectify some of this inefficiency that occurs, it's this 30% that's really where it's at. So recently I had a student, Vanessa Asseri, and she did her PhD thesis trying to find out how much in the way of nutrients are in the immune system and what the balance of those nutrients are in the immune system relative to what's in the rest of the body. So her thesis project was really to carve out the immune system out of a chicken and see how much amino acids are there. We also looked at some vitamins and minerals. I'm only going to talk today about amino acids, relative to other parts, other things that are going on in the animal, to try and get some feeling for the balance of nutrients that are needed for an immune response, as well as the amounts. And so that's what I want to dive into a little bit right now. So what she did was she injected E. coli into the bloodstream, dead E. coli in the bloodstream of some fast-growing chickens. They're about 1 kilogram in size in this experiment. And then she killed bunches of them at time 0, 4 hours, 18 hours, 24 hours, and 48 hours This is typically the period that we call the acute phase period. It's when the innate response is really very robust, and it's the predominant part of the immune response. She also looked out further on at 3 days all the way out to 21 days, and that's when the adaptive system is kicking in. That's when the lymphocytes are proliferating and starting to contribute to the immune defenses, and B cells are starting to make lots of antibody and things like that. That takes at least 3 days before you can even measure much of it, and it's usually maximum around 7 to 10 days after a novel challenge. So she, and I'm not sure, I'm trying to switch back and forth so I only offend half of you in any one given time. So she pulled out various immune tissues, and chickens are a little different than mammals. Chickens are hatched without hardly any lymph nodes. They only have a couple lymph nodes, some of the eyes and a few other spots, but they have almost no lymph nodes, unlike mammals do, where we got them everywhere. So we don't have to worry about lymph nodes. Chickens actually build a lymph node for each infection that they get. If you inject something in the wing of a chicken, they'll build a lymph node in the wing. So you can actually then carve out that lymph node that was built just for that chicken's one response. Completely, it's a nice model for doing nutrition. You just can't do this in a mammal. So anyway, she pulled out the lymph node that was formed after it was formed. She also pulled out the bursa, which is where B cells develop, thymus, where T cells develop, the liver, which contributes to some of the protective proteins, like that's where most of the complement's made, spleen, and the bone marrow, which is where a lot of the immune cells are being manufactured all the time. Then she isolated the different types of leukocytes out of each of these tissues, different types of T cells, B cell, innate cells. She also measured levels of secreted proteins ranging from immunoglobulin and including immunoglobulin specific for the challenge that she gave them, the E. coli, as well as all the rest of the immunoglobulin, and then about 22 different protective proteins that are called the acute phase proteins, and that would include complement. And she also looked at other organs like muscle and liver, where some of these things are going on. So we separated all these different cells, and we had the cells in tubes, and then we sent them off and got amino acid analysis done. We got some trace minerals done, a few vitamins done, and we now know the nutrient content of about 8 different cell types and 22 different proteins, and on and on and on in the immune system. And this has been published, and I'm not going to show you all the data. I encourage you to go look at the publications, and you're going to have to dig all the way back to the appendixes if you really want to find out what's going on in a T helper cell versus a T cytotoxic cell or something like that. But that's there. So I'm going to re-aggregate all this data that she has put together to give you the helicopter view of what's going on. So this is an unchallenged bird. I call that maintenance. That's where the immune system is just at maintenance. It's not really doing anything other than just looking. And this is during the first 48 hours of the immune response. That's when the inflammatory response is very strong. This is out at 5 to 7 days. That's when it's predominantly the lymphocyte-mediated responses. And I'm only going to show you the amino acid lysine. And I chose lysine randomly. It wasn't randomly. I chose lysine because it's used in the ideal protein system. I didn't choose it because I thought it was especially important for the immune system. In fact, we know it's not. Lysine deficiency is not an amino acid deficiency that really hurts the immune system very much. There's other amino acids that'll fall in that category, but not lysine. So I didn't pick it because it's important for the immune system. I just picked it as a reference amino acid. We measured them all, and we published the levels of all of them. So if you want to go find your favorite amino acids, you can look at that. So these are the different time periods. These are the different places we looked. Starting down here, this is the amount of lysine that's in all the immunoglobulin that's in the body. The systemic components of the body would not include immunoglobulin that's along the intestines or the lungs, but the rest of the body it does include. This is the amount of lysine that's in the protective proteins, things like complement, C-reactive protein, alpha-1 glycoprotein, those sorts of things. This is the amount that's in the various leukocyte pools, peripheral leukocytes and splenic leukocytes, and some of these pools are so small that it doesn't show up on the slide. But you can see that a lot of the lysine is in circulating proteins, antibodies and the protective proteins. The cell is quite a bit less. During the acute phase response, the amount of lysine that's in these protective proteins increases considerably. The amount that's in the cells also increases, but the real increase is these protective proteins, these acute phase proteins. Let me get a few. This is one of these. I hate this slide. I hit the button so many times. So these are the actual amounts of milligrams. This is per bird, but these birds rate about 1 kilogram. So the total amount of lysine within the protective cells and proteins of the immune system basically went up by a little more than doubling during the first 24 hours of the immune response. So you think about it, whatever you think of the immune system, and this is a question I always ask my immunology students on their qualifying exams, is how big is the immune system? You know, is it as big as a liver? Is it as big as a lung? How big is it? And they all think it's like 5 livers. Immunology is so important to them. But it turns out the immune system is really not that big. So it's a pretty small system relative to other things. But it more than doubles. So that's a pretty quick increase. And then this is out during the adaptive phase. And you can see now there's more total immunoglobulin. And now this little orange bar here is the amount that's in specific antibody. So that's a couple of milligrams of lysine in antibody. And to put that in perspective, on a daily basis, that's about the amount of lysine I calculate that I use to grow my eyebrows. So not a lot. One of the things, though, that I think is intriguing here is that all this lysine that accumulates in the protective proteins becomes available for the adaptive response because these protective proteins are degraded. They're really only around if the infection's resolved, they disappear. They're degraded, and that lysine's freed up. And so actually from a requirement point of view, the requirement for the adaptive immune response can be completely fueled by the innate response, the decay of the innate response. Now, if the pathogen's not thwarted, most infectious challenges are resolved within 24 to 48 hours. According to the human infectious disease experts, about 95% of all of our infectious challenges are resolved within 48 hours by the innate system. The adaptive response still occurs, but it's occurring separately. So again, the expense for most infectious challenges and most pathogens is the initial innate response, and then this can ride on the decay of those nutrients that were collected. And this is true for all the amino acids we've looked at. Okay, more numbers. So this is putting it all together and putting it in terms of the amount of lysine within the professional immune system, the white blood cells and protective proteins. At maintenance, about 4/10 of the body's lysine is in the immune system. During the early response, that goes up a little bit more than double. In the late response, it's less than it was there. This is the amount of lysine that we can attribute to the liver's protective function. Not its homeostatic functions and supporting growth and all of that, the production of all these protective proteins, the acute phase proteins, and some other things that it exports, like some antioxidants, enzymes, and things like that. That's the amount of lysine that the liver is doing. Plus, the liver actually hypertrophies. It increases in size by about 6% in the first 48 hours of an infection. Every other tissue other than the spleen decreases in size, but the liver and one immune organ, the spleen, increases its size. It's anabolic. So if we add in the amount of lysine that the liver needs for its protective components, with that up here, we come down to about close to 3% of the body's lysine is being used for protection. Again, more of it by the liver than by the white blood cells. And then during an early response, that goes up 4.4% and 3.1% for the adaptive response. So now for me, finally, I'm starting to have some numbers in my head. Before, I would just had these ideas that all these nutrients were flowing to the immune system, but I really had no idea. I just assumed it would be all those nutrients that are now not being used for muscle growth, but it turns out it's not. In her work, she found that muscles, the decrease in lysine uptake by muscle provides 3 times more lysine than we can account for being used by the protective responses. So that's pushing a lot of lysine out to the immune system that now can utilize it. And there's a lot more than it's needed, and a lot of that's degraded and used, catabolized and used for energy. That's been known for a long time. Okay, this is a simple slide. I spent 5 years and over $500,000 on a project where we made B-cell and T-cell knockout chickens using CRISPR technology that you heard about a couple of talks ago. We got them on the ground. My goal was to use them to find out how expensive it was to use B-cells and T-cells. If they don't have any, and we have some that do have some, we can answer that question. The bottom line is we couldn't measure any cost of B-cells and T-cells. We tried, ran lots of experiments, we failed. And win some, you lose some. But you know what? We started this project before I saw that data I just showed you on the previous slide, and really when you think about what's on that previous slide, we couldn't measure those small amounts, tenths of a percent of lysine that's going in for antibody production and things like that. We should never have done this project, let's put it that way. But it does confirm what we saw using other techniques, I guess, other avenues. So again, the real change in my thinking in the last 4 or 5 years is really the liver. To me, the liver is the real cost of an inflammatory response, and I think there's two costs. I already told you about the lysine cost, the nutrients that are needed to make these protective proteins, and by the way, most of them have a vitamin or a trace mineral as a cofactor, a lot of zinc cofactor. Turns out that when we use a metric like zinc or iron, the liver's more than 3 times the amount of nutrients that are being used by the liver for the protective proteins than by the white blood cells themselves. So it amplifies that conclusion. So the costs are twofold. One, using the nutrients, but two, the normal homeostatic things that go on in a healthy situation, which are there to maintain nutrient homeostasis, to export nutrients to growing tissues, to export vitamins and things like that in a form that can be taken up by growing skeletal muscle and all that kind of stuff. These things all slow down. The liver diverts its cellular machinery to produce all of these protective proteins. Again, ceruloplasmin, mannan-binding protein, C-reactive protein, alpha-1 glycoprotein, complement, on and on and on. And so these functions decrease and those increase. And I went back and looked at some of the force-feeding literature that was done where people infected animals and then force-fed them. And if you look in the results, there's always a statement about how the liver was huge and it was full of fat and all this kind of stuff when they force-fed the animals. And it turns out, and this is a project we have going on right now, it turns out if we force-feed our chickens after we challenge them with an inflammatory thing like an LPS or an E. coli, they can't handle those nutrients. They come in, but they don't go out. The lipid just sticks there. And it can cause necrosis of the hepatic cells. So I now believe that the decrease in food intake that happens is partially to spare the liver for having to process all those nutrients that are coming in, because it now has to be the biggest part of the inflammatory response. Its responsibilities are protective proteins, and we know those are very, very protective. Without them, we have major problems. So the liver's priority when there is a challenge, an inflammatory challenge, is to aid the immune defenses and not to contribute to homeostasis. And again, my thinking is that that's a trade-off. It can't do both. It can't process nutrients and do its job for the immune system well at the same time. What am I doing? I'm hanging in there. For some reason, I'm not getting, maybe I'm aiming this the wrong way. There we go. Skeletal muscle, we know skeletal muscle, in a healthy situation, we have lots of synthesis and not as much degradation, and in a challenge situation, protein degradation goes up considerably. The amino acid balance that we feed for is highly related to the amino acid balance of the skeletal muscle. It's not perfect ratios, but it's pretty similar. And we also know that in a strong inflammatory response, most of the amino acids that become available for the immune system and the liver are coming from skeletal muscle. So we looked at the ratios of amino acids that are in skeletal muscle versus the amount that's in the immune system, plus the protective proteins and the hypertrophy that's going on in the liver, because we had all that amino acid data. So these numbers are the ratio that's actually in skeletal muscle versus the amount that's in, let's just call it the protective immune system. So for arginine, ratio 0.81 that we see during the first 24 hours means that there's more arginine in skeletal muscle than there is in the immune system from a proportional, from a balance point of view. But now you look at the cysteine, there's about 90% more in the protective proteins than there is in skeletal muscle. So this, to me, would be a bottleneck. We're not the only group that's found this. In INRA in France, about 3 or 4 years before we got this information, they did a somewhat similar experiment looking at the liver versus skeletal muscle and acute phase proteins and found cysteine to be the biggest bottleneck for the inflammatory response in rats. But cysteine is known to be really highly enriched in cells, in proteins that have to have a lot of integrity. They have to be able to withstand proteolytic attacks, oxidative attacks. And protective proteins are at sites of infection where there's lots of proteolytic enzymes being released by the immune cells themselves, but also the pathogen, because there's a war going on there. Free radicals are being produced by the immune system to kill the pathogens. So cells that we know need to act at sites of infection have to have lots of cysteine molecules to give them the structural integrity to maintain activity in those pretty dangerous environments. Among some of the others during the acute phase response, threonine pops up. I've been concerned about this cysteine, so we started looking at conversion of methionine to cysteine during the inflammatory response, because as I just mentioned, liver function, we find a lot of those homeostatic liver functions are now not happening very well. And we found that the conversion of methionine to cysteine in the chicken during inflammatory response is about a third, the flux is about a third as much as during a healthy situation. And many times when we add methionine to the diet, we're adding methionine to supply cysteine. In broiler chickens, that's always that way. And even in pigs, when I formulate pig diets, many times what's triggering the inclusion of methionine is cysteine. So I worry whether they can really rely on that in a situation where the animal's been challenged by an infectious disease. During the adaptive response, out here, cysteine is also important because it's really highly enriched in immunoglobulins. Another one's proline, and another one's valine that popped up that would be real critical bottlenecks to think about if we're going to formulate a diet specifically for infection and to support the immune system. These are the ones that pop up. Okay. If you read the literature, the human literature, there's been something that's been described since the early 1000 AD in writings, and that's something called, translated these days into a lipid excretion during infections. And it's an observation that's been used for diagnosis of infections for hundreds of years, and that is more lipid in the stool. That's a common thing. You look at many infectious diseases, that's one of the symptoms, is lipids in the stool. Well, there's some nutritional signals there when you see lipids in the stool. That's a malabsorption. And that's been studied pretty vigorously in mice. And it turns out the intestines obviously have absorptive functions, and they also have barrier functions to keep microbes out. But during an inflammatory challenge, there is a switch over, a decrease in absorptive functions and an increase in barrier functions. Changes in the numbers of tight junctions in the cells, changes in expression. Changes the rate of replication of the supply enterocytes in the crypts, shortening of the villi, less absorptive surface. A whole variety of different changes have been described. This happens during intestinal inflammation, but it happens just as much if the inflammation is systemically, nowhere near the intestines. And it's been shown that it's being caused by these pro-inflammatory cytokines that I talked about previously, providing a signal for the gut to switch over to a more barrier phenotype. So we've examined, this has been examined a fair amount in pigs. These are a couple studies that came out in the last year. This one looking at changes where they injected LPS, and this study I think was actually into skeletal muscle, and then they measured villi lengths and crypt depths and those sorts of things. This is just one table out of many in this paper, and they described a change in phenotype of the intestines is really similar to the same thing you would see during a severe intestinal inflammatory challenge. They didn't actually measure absorption of nutrients, but we ran an experiment recently where we did that. We fed healthy chickens as well as those we induced an inflammatory response and measured ileal digestibility of various nutrients. And that's what I have here. And in some cases, the ileal digestibility went down. Methionine was decreased by 8%, fat by 19%. So this is that fat excretion, this steatorrhea of infection or inflammation that's been described for so long. 20% decrease in fat absorption during the inflammation. This injection was systemically. We injected it into the blood. So this is not a result of inflammation in the intestine. The inflammation is elsewhere. All of the minerals we looked at were decreased. Calcium by 10%, iron by close to 40%, retinol by close to 50%, the carotenoid lutein by 65%. So then my student, Vanessa DeGroris, she had the idea that what we ought to do is formulate a diet that has 8% less methionine and 19% less fat and less calcium, et cetera, and feed that to healthy chickens and see how fast they grow. And then compare it to those that got their normal diet that met the requirement for everything. And what she found was they grew 9% slower. So to me, this malabsorption due to an inflammatory response decreases growth rate by 9% just from malabsorption. The inflammatory response does not need to be in the intestines. It can be anywhere. So getting back to my figure over here, where we're looking for 30% decrease in growth that's due to things other than food intake, I already described to you that the actual amount of lysine that's being used for the immune system and the protective proteins in the liver accounts for about a 4.4% decrease. And the metabolic efficiencies includes that mismatch between the high enrichment in cysteine versus what's coming out from skeletal muscle, because to get that extra 90% cysteine, you have to release 90% more skeletal muscle. It also includes some of the amino acids that are used for gluconeogenesis, some other inefficient things that happen during inflammation that I haven't had time to go into. We've measured the amount of energy that's used just both by the cells, but also for fever that happens. That accounts for about 3.5%. I just described the digestive efficiencies, that's about 9%. So add that up, we're about 25%. I'm looking for 29%. I retire in 4 years, that's 1% per year. So anyway, we're getting close to this. When I started this business, I thought it was all here, all in the cells and protective proteins, and it turns out much of it's not there. Okay, just to put this in perspective, because when I talk to non-nutritionists, like when I talk to immunologists, they don't understand what a 15% decrease in lysine or whatever, all that. They don't understand all this nutrition stuff. So I try to convert this into something they can understand. The immune system, when it's idling along and doing nothing, has about the same amount of lysine in it as 15% of an egg, or about 5% of a pectoralis muscle. When it responds, and when you include some of those inefficiencies I talked about, it's utilizing about as much lysine as about 40% of a pectoralis muscle, or 1.2 eggs, from a material point of view. Well, we know that in our inflammatory model, if we do it in a laying hen, they don't lay an egg for about 2 or 3 days. They stop laying, so that makes sense. So this is just calculating the amount that's being diverted for these other things. Okay, I'm going to spend just a minute on priorities for nutrients. It's an area I've been interested in for a long time. We know that different types of cells have different priorities for nutrients. For example, we know when food comes in, nutrients come in, brain gets it pretty well. It's hard to starve the brain and the heart. Reproduction in some species will continue reasonably well. And then lower priority tissues are things like muscle, and the very bottom of the barrel is fat. And of course, during starvation, the high priority tissues dominate the carcass, and the low priority tissues are disappearing. And this is mediated by a number of different mechanisms. nutrient transporters, various ways to store nutrients, and the combination of those two. In the case of amino acids, it's predominantly by the nutrient transporters. Same is true with glucose. And there's usually, for any nutrient, let's say, for example, lysine, because I've been talking about it a lot, there's a transporter family called the cationic amino acid transporter. In chickens, there's 3 isoforms of that. 2 of them have a low Km and a low Vmax. They're able to transport lysine at really low concentrations, and that's expressed a lot in high priority tissues like the brain and the heart. Another one, Cat2, has a really high Km and a really high Vmax. That's in muscle. It can bring in lots of lysine, but only when it's at high concentration in extracellular fluids, like after a meal. So it's the lower priority, but high transport rate. Same thing goes on with glucose and many different nutrients. So we've measured the priority of different tissues, for example. This was done by Brooke Humphrey, who's now a pig nutritionist. And this is part of his PhD work. But basically, I don't have heart and brain on here. They were up around 5. But some tissues like the bursa, high, liver's more intermediate. Here's the heart, which was in this experiment more intermediate. Gastroc, the thymus is way down here. Pectoralis was the lowest numerically of any of them. So this aligns with what we typically would see with starvation. We would see a maintenance of the bursa, liver, heart, and diminution of the muscles. And he did quite a bit of work looking at this in lots of different cell types under lots of different conditions. And I think it's really interesting stuff, but I don't have enough time to get into it in a lot of detail. For me, the interesting take-home is that macrophages and B cells have a really high priority, and T cells have a really low priority. In fact, T cell priority is right about where skeletal muscle is, unless they've been activated by a pathogen. Then it skyrockets up. But normally T cells are there. So nowadays, when we're looking at the immune system getting the nutrients it needs for responses, we focus a lot on T cells because they're on the low end of the priority scale. Move this along. So this summarizes it from a priority point of view. We've got T cells at the bottom until they get activated, B cells resting pretty high priority, activated very high. Macrophages and the production of acute phase proteins is always very high, and in most cases, in most of the experiments we've done, it's higher than the brain or the muscle. I'm sorry, or the heart. So this sort of tells us who to worry about during an immune response. But obviously we want muscle. Well, one thing I've noticed, and I've actually caused to some extent, is that our LPS model that we've used, our E. coli injection model, is not as robust as it used to be 30 years ago when I was starting this work. So I went back, and I've worked with both broilers and layers, and I plotted the decrease in appetite, the change in appetite that has occurred. So this is the amount of anorexia, so the higher the number, the more anorexia. These are laying hens since 1980 up to more currently. There's been a slow drift downwards of the amount of anorexia that happens during inflammation. In broilers, there's been a quick drift downwards. Now we get about a third as much anorexia as we did 30 years ago. And I know why this has happened, because I was involved with it. I've consulted with all the broiler breeder companies and all the pig breeding companies and helped them design ways to do selection against anorexia during infection. This is before I knew about this liver stuff I just talked about. And they consciously bred. It wasn't one of the major driving selection factors, but they bred for decrease in anorexia. We also know that even before they started doing that, there was still a slow downward drift. And I'll show you a little data on that in a second. That probably was just secondary for breeding for fast growth and efficiency under normal production situations. So is this good? Well, I'm not 100% sure it is good. It could, I mean, it's good if you have healthy animals. You don't have to worry about it too much. It's good if the inflammatory challenges are pretty minor, but maybe if it's a major, huge problem like PRRS or something like that, it may not be good at all. Come on, there we go. So one of my old students, Doug Corver, who's up in Alberta, he has some broiler, Arbery acre broilers from 1957 that were then frozen in time and no longer randomly bred. And he has some that were bred for fast growth and efficiency to 2007, and now they've been randomly bred since then. And we measured the amount of high priority versus low priority lysine transporters or cationic amino acid transporters in these birds following an LPS challenge. And skeletal muscle now has about 2.3 times as much high priority transporters as it used to. In pectoralis, gastroc, about almost twice as much. The immune tissue really hasn't changed at all. We've actually, over time, I think, for breeding for lots of muscle and for high feed efficiency, we have increased the priority of muscle relative to where it used to be many years ago. Is this a good thing or a bad thing? Well, I don't know. Most often it's probably a good thing, but I'm sure there could be some situations where it results in increased mortality due to infectious challenges. How am I on time? Because I was really not given a time to end, because there is no ending time on here. 5 more minutes? Is that okay? Okay, good. Because I want to talk about some mitigation strategies. The biggest in mitigation strategies, don't let there be inflammation. Okay, pretty straightforward. Sanitation, vaccination. Although most good vaccines cause some inflammation. The better, the more protective the vaccine, the more inflammation you get when you give the vaccine. So there is a trade-off there. So you have to evaluate that versus the amount of protection and your probability of getting the pathogen into your herds. Spend the money for the insurance or not spend it, you're going to have to do those calculations. But just realize there is a cost of vaccination due to the inflammation, but it can protect you against an awful lot of things. So that's an important consideration. We also get inflammation coming from dietary components, physical irritants like silica. Some particle sizes of silica can cause some pretty severe inflammation. Plant defense molecules. Plants defend themselves against being eaten by insects or mammals by producing things like lectins, soybean agglutinin. It's highly inflammatory unless we denature it by heating it. Mannins, for example, when we've done some experiments on those, can be pretty inflammatory, and there are lots of different food types. Saponins are another one. And again, they're produced primarily and loaded into nutritionally important parts like seeds and nutrient storage organs in plants as a way to defend themselves against predation. And those are other ones that we need to worry about that don't fall into the pathogen side of things. And then next, many people have been looking at nutritional anti-inflammatory things because we know plants not only have inflammatory things, but they also have anti-inflammatory things. And this is a big area in human nutrition. I've listed a number of them here that have been shown to have anti-inflammatory properties, things ranging from oil like this should be omega-3 fatty acids, like fish oil, several non-essential amino acids, vitamin E, vitamin D, lutein, a number of them. One thing that really surprises me when I look at this list is rarely that any of these are even essential nutrients. And the few that are there that are essential nutrients aren't structural components of things. You know, they're not amino acids being used to make proteins. They are more like hormones, like vitamin D. And vitamin E has some hormone-like properties too. So these are the ones that tend to be anti-inflammatory. There's other nutrients that are inflammatory, but we have both sides. And we might be able to use these to decrease inflammation. And I've worked on that for a number of years. For example, we've done a lot of work with fish oil. Let me go back, if I can get this to go back. There we go. And a lot of work's been done on fish oil. Over 2,000 papers have been published on anti-inflammatory effects of fish oil, especially in mice. The mechanisms are really well known. I don't want to get into them. But we also know, and this is Kevin Fritchie's work from University of Missouri, that, and each of these are individual study, that fish oil, if you feed it, it's anti-inflammatory, but there's lots of infectious diseases that increases the mortality to those infectious diseases because there's many diseases where inflammation is part of the protection. And so there's a trade-off. You might be helping yourself in one spot, but you might be hurting yourself in another spot. And I wish it was that simple. Well, actually, here, this is a fun experiment we did a while back. I did this with the broiler integrator. We had 672,000 chickens that we fed a regular diet, and 672,000 chickens we fed 3% fish oil. We followed them all the way through slaughter. We got their condemnation records from the USDA inspector, and there was a decrease due to the fish oil of several different condemnation categories, things like airsacculitis, cellulitis, septicemia, toxemia. These are all caused by inflammation, and it helped with those. Inflammatory processes of the skin, inflammation, really helped with that, decreased incidence of about 70%. This is a viral infection that causes tumors. The category is called leukosis. It's actually caused by Marek's disease virus. Increased by about 50%. 50% more tumors in these birds because we gave them an anti-inflammatory compound. It turned out that there was a low incidence of this is a percent increase, low incidence of this and high incidence of this one, so they loved me. But man, if I'd have gone in there on a year when they had a lot of Marek's and not so much skin inflammation, they'd have booted me out for the rest of my life. But it's more complicated than that. People in Europe have done similar types of things, and they never found the same results with fish oil that we were finding here in the United States, even with the same strain of broiler birds. And we started looking into that, and one of the main differences is we feed corn and they feed wheat. And there was some indication out of the mouse literature that lutein is anti-inflammatory, especially when there's a lot of omega-3s in the diet. And so we started looking at this interaction, and we found that to be true in our chickens. We feed lutein alone or with fish oil, you get a much bigger effect of fish oil when there's lutein. Each of them independently a little bit of decrease in inflammatory response, but put them together, you get a synergism in decrease. And then one of my students worked this out mechanistically, and that's published in Journal of Nutrition about 10 years ago, so I don't want to go into it, other than to say that there's actually a receptor for omega-3 fatty acids and a receptor for lutein. Those receptors dimerize, they go to DNA, and they change expression of things. You need them both to get a good change in expression. There was also some indication that lutein interacts with vitamin E, and so we looked at that, and we found, yes, it does. Vitamin E and lutein alone is decreases inflammation a little bit. You put them together, and it increases inflammation. So they have a complete opposite effect. And the mechanism for that's been worked out, too. When lutein binds to the RXR and when vitamin E binds to a different PPAR than omega-3 fatty acids do, it binds to different sites on DNA, and it increases the expression of pro-inflammatory cytokines and inflammation. This, to me, is scary because you think you're doing one thing because we do these experiments in isolation, just vitamin E or just lutein or just fish oil or whatever, or just the anti-inflammatory compound of the moment. You get one result, and you start putting them together, and you get sometimes the opposite results, sometimes synergistic results. And it's difficult to predict because we don't know all these mechanisms yet. So again, sometimes you take something that increases or decreases inflammation, another one decreases inflammation, and you get an increase in inflammation. We can't predict when that's going to happen for most of these immunomodulatory nutrients. So we have a lot more work to do, I think, before I can really feel comfortable using a lot of immunomodulatory nutrients specifically to do things that we think we're doing. We really need to look at it in more than just simple experiments. And so I still take money and do this research, but I really think you need to give me more money because I think to really do it safely, we're going to have to do a fair amount of research. And I think I'm going to end on this one. I had one more small topic I wanted to cover, but I think this gives you all the major points, and I'll stop there. And if there is time for questions, I'd be happy to take a few.